Time for Chapter 11. Chapter 11 is all on muscle physiology, so looking at how our three different types of muscle not only work, their individual characteristics, and looking at the basics of a muscle contraction. All muscles have certain universal characteristics. So first one is that they're excitable, so they will receive some kind of signal that causes them to react. They can conduct an electrical signal. They are able to contract. So this is something that is unique to muscles. So it's one of their unique characteristics is that they will contract or shorten when they're stimulated. Muscles can also be stretched, so they're extensible, so going beyond stretch between your contractions. And then lastly, muscles are elastic, so they'll go back to their original length after being stretched. So just like when you work out in the gym, stretching your muscles, moving them past the point of their normal positioning, and then able to pop right back into their normal length. First type of muscle that we'll look at is the skeletal muscle. So these are the muscles that are attached to bones usually. Things that are characteristics of your skeletal muscle is that they have striations. So in our picture here, let's zoom in. There are alternating bands of light and dark color based on the overlap of your different filaments. So we'll talk about that later. So that causes that striping pattern to appear in your skeletal muscle, as well as in your cardiac muscle. One thing that's unique to your skeletal muscle is that it is voluntarily controlled, so we're able to consciously control skeletal muscle. So we're able to use our brain to consciously or be completely aware of where we're contracting our muscles. So we can control where our muscles are going. So kind of like I'm doing with my arm, telling my muscles in my arm how to move the pin around. However, for your smooth and cardiac muscle, we cannot tell our heart to slow down or tell our stomach to speed up digestion and mix things up a whole lot faster. That we cannot consciously control, and so it's under involuntary control. The cells for skeletal muscles are called myofibers. They can be as long as 30 centimeters. So again, this picture here. And in a second, we're actually going to go to a cartoon version, but still looking at the different components. The muscle fiber is another term for talking about the cells of your skeletal muscle. With your skeletal muscle cells, they have certain terminology that's specific for that cell. So instead of your normal vocabulary terms for looking at cells, for your muscle fibers, they have special terms for their different features. So on the outside of your muscle fibers, instead of having just calling it a plasma membrane, that outside wrapping, which is still just like your regular plasma membrane, just special name, is called your sarcolemma. Inside your sarcolemma is your sarcoplasm, so just like your cytoplasm of a regular cell. Part of your sarcoplasm contains your myofibrils, so these are most of the space that are inside your sarcoplasm, so these are going to be the ones that help with contracting your muscle and shortening of the muscle fibers. Other things in the sarcoplasm, you have glycogen. So if you remember from earlier, glycogen being the storage molecule for glucose and giving it the energy to make ATP. Another component is myoglobin. So just like hemoglobin in our red blood cells, our muscle cells have their own source of oxygen. Again, oxygen needing for aerobic respiration to be able to make ATP. So both of these providing the glucose and the oxygen to make ATP inside your muscle fibers. Other features of your muscle fiber, so still talking about your skeletal muscle cells, particularly for just your skeletal muscle, they have multiple nuclei. So during their development, these myoblast cells will converge into each other, so they'll fuse together to form your muscle fiber cell. When these stem cells fuse together, the cells fuse, but not the nuclei. So because the nuclei don't fuse together, you end up with multiple nuclei in this cell. So just like your osteoclast in bone tissue have multiple nuclei because of the fusion of cells, same thing with your skeletal muscle fibers. Associated with your muscle fibers are the satellite cells. So they're unspecialized myoblasts, so they haven't been differentiated to turn into our muscle fibers. These satellite cells help to regenerate damaged skeletal muscle tissue. So, for example, as you're working out and you make those little tears in your muscles as you're, lifting weights, those cells help to repair your muscle after doing so. With your muscles needing a lot of ATP to be able to power up the muscle contraction, and we'll see this in a little bit, there's a lot of mitochondria in your muscle fibers, and they're actually a lot bigger than a normal mitochondria, but again, needing a lot of ATP in order to have the muscle contractions. Last specialized section of organelles and features of your muscle fibers is that there's your sarcoplasmic reticulum. So just like your endoplasmic reticulum in a regular cell, your sarcoplasmic reticulum, instead of just being part or wrapping around your nucleus, your sarcoplasmic reticulum wraps around your myofibrils. So they're kind of like a cage on the outside, and I'll show you in a picture in a little bit. With your sarcoplasmic reticulum, important parts for it is that it has these terminal cisterns where it acts as a calcium reservoir. Again, calcium is needed for your contraction. Show you this in a little bit. So these terminal cisterns, which are part of another feature called a triad, these terminal cisterns will release calcium once they're activated to do so through a action potential. And the calcium is then released in order to activate your contraction. Associated with your sarcoplasmic reticulum are your T-tubules. So these are tubular-shaped infolding. So like you have your plasma membrane, and then there's a infolding where this folds in of your plasma membrane to make a tube across your cell. These T-tubules are where your action potentials are going to travel. So your action potentials start at your neuromuscular junction where they're getting the nerve signal to cause the muscle to contract. Those action potentials leave that junction where the synapse from the neuron or from your nerve cell activates the muscle. They travel along your sarcoplasm, sarcolemma, so your cell membrane for your cell, and then they'll move into the T-tubules to be able to activate the sarcoplasmic reticulum to release the calcium. So your sarcoplasmic reticulum on both sides of your T-tubule, so you have a T-tubule in the middle, a terminal cistern of your sarcoplasmic reticulum on the sides of that, so there's three parts of that. They make up the triad, so the T-tubule and the two terminal cisterns that are associated with it. So here's a picture of everything. So here's a skeletal muscle fiber, again, a cute little cartoon version of it, but it still works. So on the outside in yellow is your sarcolemma, again, your plasma membrane for your muscle cell. Your cytoplasm of your muscle fiber is your sarcoplasm, so kind of showing that in the kind of little beige color here. Important parts for the skeletal muscle to actually function. So we have our myofibrils here in the pinkish and red colors. So with your skeletal muscle, because their main function is to contract, so getting these myofibrils to work together, the nuclei are pushed to your outside of your cell. So right up against your sarcolemma. So around bundles of your myofibrils, you have your sarcoplasmic reticulum, so that's the cage-looking structure in blue. The triad here, so we have our terminal cistern, another terminal cistern running in the middle of that is your T-tubule or transverse tubule. So infolding, see these little holes where your action potentials traveling along your sarcolemma will travel into the cell and be able to activate these terminal cisterns of the sarcoplasmic reticulum to release the calcium. Stuffed in between your myofibrils are your mitochondria, so very large mitochondria to make sure that you have enough ATP to have your skeletal muscle contractions. And lastly, second to last thing I'm pointing out, you have your myofibrils. Again, these will create a greater level of overlaps to cause your skeletal muscle to contract. There's two types of myofibrils. Inside your myofibrils are your two types of myofilaments, so we'll talk about those in a second. And last thing I want to point out is that your myofilaments are arranged in a way that causes certain structural features to be pointed out in your skeletal muscle. So they're on the drawing, so I'm going to point them out anyway. So some of your myofilaments are anchored. They're actually all anchored to the Z-disc in some way. This is just a structural element to allow for the linking of anchoring of your myofilaments. And then with your overlapping of your myofilaments causing the striations in your skeletal muscle fiber, you end up with your more darker staining A-band and, not staining, more darker looking A-band and your lighter looking I-band. First type of myofilament is your thick filament. It's made of a protein called myosin. Your myosin molecule is shaped like a golf club. So here's the head portion, and this would be like the shaft of your golf club. That's called the tail in our case here. So these myosin proteins are arranged in like a helical shape around kind of like a tube. The head's pointing towards the outside. With your with your thick filaments, your heads point away from this middle bare zone. So where there's no overlap with your thin filaments, there's no need to have that head that's going to bind to the thin filaments, and therefore they're just not in that bare zone. So the tail regions being here and then pointing to your Z-disc. So what we can't see in this picture, you'd have a Z-disc on this end and a Z-disc on this other end. The other type of filament is a thin filament. So it's made up of your actin filaments, actin protein. So these red balls here are actin. And associated with your thin filaments are tropomyosin and troponin molecules. So these two molecules are regulatory proteins that help your muscle cells regulate whether or not they're going to contract. So here's why we need the calcium reservoirs on your terminal cisterns from your sarcoplasmic reticulum to be in your muscle cells. So once they're activated, releasing the calcium. Calcium binds to your troponin, so that's this little yellow structure here. Binds to that. This tropomyosin blocks the binding sites on your actin subunits. So when that calcium binds, that tropomyosin removes off of those active sites on your actin subunits and allows for those myosin heads to bind. So basically it kind of acts like a gate, and your calcium is the key. The last type of filament that's involved with your muscles are your elastic filaments. So like their name says, they're elastic, they're stretchy. They are also anchored to your Z-disc, so your thin filaments here, kind of orange color, and our elastic filaments that are also anchored to our Z-disc are in the green color here. They run through the middle of your thick filaments, so it helps to keep it in place, keep it kind of in the center portion of that myofibril. So it helps to anchor it to your Z-disc, so keep everything in place. So it helps to prevent overstretching, but also provides recoil, so again, it's elastic, so it helps to spring stuff back in together. So I'm going to point out something here, since we have all the little pieces together. So the I-band is your lighter striation or stripe for your striations, where you don't have overlap between your thick and your thin filaments. So just in this region, including the Z-disc, is your lighter I band. Your darker A band is where there's overlap, and so therefore it ends up looking darker and causing that striping pattern on your striated muscle. Okay, last thing, just focusing on our myofilaments and their components. I said this earlier, but I can say it again. The myosin and actin on our thick and thin filaments actually do the work of causing contraction. So these are our contractile proteins. The regulatory proteins are our tropomyosin and our troponin. So again, calcium released from the terminal cisterns of your sarcoplasmic reticulum bind to the troponin. And when the troponin is bound with calcium, it changes shape, so it goes through a conformational chain to move the tropomyosin off of the active sites on actin, allowing for the myosin head to bind and cause the contraction. Quickly going over the striations again, it's due to the overlapping of your thick and thin filaments, leaving the stripes made by the A band, which is your dark band, due to the overlap of your thick and thin filaments, your I band where you only have your thin filaments. Also, your Z disc is located in that area. So I band and A band. So important functional unit of your muscle cells are your sarcomeres. So this is a segment of your myofibril from your Z disc to a Z disc. That's your functional contractional unit. So when this unit shortens, it helps to shorten the muscles. So they all work together to shorten your muscle fibers, and then therefore all your muscle cells working together to shorten the entire muscle. When your Z disc to Z disc sarcomere shortens, doesn't shorten due to the change in length of the filaments. It shortens because of the overlap. So more overlap shortens the sarcomere. Moving on to the structure of skeletal muscle. So starting from the entire muscle itself, your muscle is also enclosed, just like a lot of your other tissues and organs are enclosed in a type of connective tissue called a epimysium. Inside your muscles, you have bundles of muscle fibers called a fascicle. Fascicle also has its own little wrapping called a perimysium. So think epi is very large, like something that's totally epic. And then peri is just something that's around. Making up your fascicle are your muscle fibers. So these are your individual muscle cells. So these muscle cells have their own, again, your sarcolemma, which is your plasma membrane of your muscle fibers. But they also have their own fiber sleeve called a endomysium. So think endo like inside. Inside your muscle fibers are your myofibrils. So this bundle of your different myofilaments wrapped in your sarcoplasmic reticulum is a next lower level in your hierarchy of your skeletal muscle. Inside your myofibril, you have your sarcomere. So again, Z disc to Z disc, and your functional contractile unit of muscle fiber. So when the sarcomere shortens due to increased overlap in your thick and thin filaments, causing sarcomere to shorten. They work together to shorten the entire muscle. Lastly, inside of your sarcomere are your myofilaments. So again, your thick and thin filaments that help to shorten the sarcomere by creating increased overlap. In order to signal our muscles to contract, they have to have a signal from the nervous system. So this is specifically talking about the skeletal muscle. So the nerve fiber and the muscle fibers that are innervated or given the signal to it's called a motor unit. So our neuron here in yellow is going to innervate the muscle fibers or our cells down at the bottom. So even if this motor unit had a ton more muscle cells that it activated, still by this one neuron. So just the one neuron and all of the muscle cells that are innervated by that one neuron. So this causes those muscle cells to contract in unison, so giving a large amount of power in a certain area, so either a weak contraction over a very large area, or a very strong effective contraction caused by several motor units working at once. So having another one where here's a neuron and then affecting more cells, so all these cells are going to work together to help to contract better of the muscle. So here's a prettier picture of the motor unit. Starting in your spinal cord that is located inside your vertebrae are the neurosomas, so the cell bodies for your neurons. Then the axon travels out through the nerves, eventually targeting the muscle, and all of the muscle cells that are targeted by that neuron make up the motor unit. To get the nerve signal from your neuron to your individual muscle cells, this happens at the neuromuscular junction. So think neuro for our neuron or a nerve cell, making a connection with the muscle cells. So it actually doesn't physically touch it, but very, very close to that muscle. So where your nerve fiber meets the target cell, so in a very close proximity to your target, it's called a synapse. The very specific synapse for between a neuron and a muscle is called a neuromuscular junction, or NMJ for short. Again, this is a very specific synapse type, so not like a regular synapse that's between a neuron and another neuron. This one's between a neuron and the muscle. So each terminal branch of your nerve forms its own separate synapse, so you can have several signals coming from that one neuron at the same time. So parts of your nerve fiber that are active in giving the signal, at the final end of your nerve fiber is your axon terminal, so it's a little bit swollen, kind of looks like a Hershey Kiss. And it contains these vesicles that are called synaptic vesicles. So these vesicles contain acetylcholine, which is your neurotransmitter or neuron chemical messenger that will activate your muscle cells. So when your nerve cell or your neuron is stimulated to release these synaptic vesicles will merge with the cell membrane of your neuron and release the acetylcholine neurotransmitter or chemical signal like a ligand out into the space of the synapse. So that space is called the synaptic cleft, or it's the gap between the axon terminal, so the final end of your neuron, and the sarcolemma of your muscle cell. Helping to keep your acetylcholine in that area as well as protect that neuromuscular junction, there's a Schwann cell. So these are the oligodendrocytes, excuse me. These are the glial cells that help to wrap around and protect your neurons in the peripheral nervous system. So this is one of the jobs that this cell does, helping to wrap around and isolate that neuromuscular junction. So making sure that only the signal from the neuron is causing the muscles to contract. When the nerve or neuron receives the signal to tell the muscle to contract, the synaptic vesicles will go through exocytosis, so the vesicle merging with the plasma membrane to release the acetylcholine into that synaptic cleft, so that gap between your neuron and the sarcolemma of your muscle cell. Your muscle cells have millions of acetylcholine receptors, so that's the ACH for short. These receptors will receive that ligand. And in order to help have all these millions of receptors, there's junctional folds to increase surface area. So just like your microvilli increase the surface area of your cells, these junctional folds allow for more surface area for more acetylcholine receptors. Wrapping around, again, your neuromuscular junction, you have your basal lamina, which helps to separate the Schwann cell and the muscle cell from the surrounding tissue. So again, protecting that junction. It also contains acetylcholine esterase. This is the enzyme that breaks down acetylcholine, so it prevents it from continuously signaling the muscle to contract. So remember, ACE for enzyme, and what is it breaking down? It's breaking down acetylcholine. Here's a picture of the neuromuscular junction. Of course, we're missing the Schwann cell, but that's okay. So we have our basal lamina separating out the neuron and your muscle fiber here on the outside. The axon from our nerve fiber, so getting to our terminal end. Then we have our axon terminal, again shaped like a Hershey Kiss. Filled with all these synaptic vesicles, these little green dots here. So those are filled with acetylcholine. That's going to be our ligand to cause the cause the action potential or the signal for the muscle to contract. So when these synaptic vesicles are given the signal to merge with the plasma membrane of our neuron, releasing the acetylcholine in our synaptic cleft, so this little empty space area, able to reach our acetylcholine receptors, which are purple lines here, again accreting the plasma membrane area of our neuromuscular junction through the junctional folds, giving more surface area for more acetylcholine receptors. And then all of the rest of the stuff is just your motor cell. Going back to our universal characteristics of our muscles, knowing that muscles are electrically excitable. So they get this electrical excitability trait due to voltage changes in response to stimulation. So a voltage is an electrical potential. So this is a difference in charge from one point to another. So in our case, looking at the difference between the inside and the outside of the cell. Our membrane potential, so our electrical potential across that membrane at rest, so not during the process of being stimulated, is called our resting membrane potential. And in muscle cells, it's about negative 90 millivolts. So that difference between the inside of the outside of the cell is negative 90 millivolts. So the inside of the cell is actually more negative. Part of this difference in charge is maintained by the sodium-potassium pump, so pumping sodium outside the cell, bringing in potassium into the cell, helping to adjust the ion concentrations in and out the cell. Those ions have charge, so it also helps to add to the difference in charge between the inside and outside of the cell. So here's a cute little picture version. Unstimulated cell, we have an excess of sodium ions on the outside, excess of potassium ions on the inside. You also have more anions on the inside, leading to a more negative charge on the inside. So here we have little electrodes looking at the difference in charge between the inside and outside of the cell. Difference is negative 90 volts, so this one is more negative on the inside of the cell. When the cell is stimulated, so once that acetylcholine ligand binds to an acetylcholine receptor, what happens is your acetylcholine receptor is an ion channel, so it opens up, allowing for the sodium and potassium to go down their concentration gradients. When that happens, there's a slight adjustment in the difference in the voltage of the cell. So the inside of your cell becomes a little bit less negative, so it becomes more positive, eventually reaching what's called threshold. When threshold happens, your sodium voltage-gated ion channels, shown here in blue, as well as your potassium voltage-gated ion channels will start to open. Your sodium voltage-gated ion channels are very fast at opening, so at the start of the changes happening with your membrane potential, so starting your action potential now, your sodium voltage-gated ion channels will open, allowing sodiums to rush into the cell, so they're attracted to the negative charge on the inside of the cell, as well as going down their concentration gradients. So this process is called depolarization. It's the first part of your action potential. The second part of your action potential is repolarization. For repolarization, the charge on the inside of your cell has now become positive. So actually ignore this negative 90 millivolts. Forgot to change that. Okay. So the inside of the cell is more positive. So I forgot to change a lot of this. Okay, inside the cell is just more positive because all that sodium that rushed in. So with your repolarization, okay, I fixed it. I just copy-paste from when I was making the figure. Okay, so during repolarization, you have excess of sodium ions on the inside of the cell, so our little blue dots here. You have still an excess of potassium ions on the inside of the cell because your potassium voltage-gated ion channels are about to open, so that's your top of your peak for your action potential. So think of depolarization as one part of the peak, repolarization is the other part of the peak. So with your repolarization, your potassium voltage-gated ion channels finally open, allowing potassium to rush out the cell, so going not only down their concentration gradient, but attracted to the more negative outside of the cell, as well as being repelled by the more positive charge on the inside of the cell. So moving potassium out the cell. So depolarization, sodium rushes in. Repolarization, potassium moves out. So this quick change in voltage, so your depolarization and repolarization, is called a action potential. So your resting membrane potential is just when your cell is waiting to be stimulated. Your action potential is a very quick event once your cell has been stimulated. So your quick depolarization, quick repolarization. And this action potential, as it depolarizes, it actually triggers the next section of the cell to start reaching threshold and start its own depolarization. So it ends up traveling along the cell's membrane until it gets to that T-tubule and then moves into the T-tubule to activate the Sarcoplasmic Reticulum to release the calcium. So this wave of excitation is called the impulse. There's a lot of YouTube videos if you want to see this kind of in action form, seeing it, actually moving. There's a lot of them that are pretty good looking at an action potential and how it triggers moving down your cell membrane.